Hi group, and welcome to Unit 4. We're going to talk about the interactions of microbes on the environment and on humans. Before we get too carried away, we have to cover a few terms, and the first term that I want to talk about is flora. And flora is basically a population of organisms that live on or within your body, okay? Some microbial flora are symbiotic and some are pathogenic. So mixed flora of different types live in the respiratory tract, the gastrointestinal tract, and the genital tract. Okay, so we have a number of different organisms that are completely normal living in those body systems, our body complexes, and we have a number of organisms that can invade the normal flora and take over and become pathogenic. Symbiosis is flora living on or within the host. Neither are harmed and both receive equal benefit. That is also sometimes referred to as mutualism. Okay? And a pathogen is flora living on or within the host that harms the host and causes disease. So some organisms are true pathogens and other ones can be opportunistic pathogens. For example, Staphylococcus aureus is a normal part of the upper respiratory tract. However, Staphylococcus aureus can cause different types of diseases, so it could be a pathogen in some circumstances. Streptococcus pneumoniae can be part of the normal upper respiratory flora, but if it's by itself or if you have a low number of normal flora organisms, Streptococcus pneumoniae can actually be a pathogen. So the hundreds of different organism types that live on or within a healthy person is called your normal flora. And like I said, the normal flora lives in places like the respiratory tract, the gastrointestinal tract, on the skin, and also the genital tract. Your resident microbial flora are organisms that colonize for months or even years. For example, we have gastrointestinal flora that is almost unchanged over our lifetime, unless for some reason we have gotten antibiotics to wipe out our normal flora, or we've had a disease that's wiped out our normal flora. Even the flora of twins or siblings that grew up in the same household have similar resident microbial flora, which is kind of interesting. Transient flora are organisms that colonize the host, but only for a temporary period of time. Now, the origin of microbial flora is when you are born, you are exposed to the environment once you are born, and because of that exposure to the environment, you start to establish your own microorganisms from coming in contact with mom, dad, your siblings, and things in your household. And like I already mentioned, normal flora can be found in several body tissues of humans, including on the skin, the upper respiratory tract, which would include the mouth, the nose, the nasopharynx, and the oropharynx. And then we have normal flora that's part of our digestive tract. That includes mostly your intestinal tract. There's really not any normal flora in the stomach, but if you look at the small intestine and the large intestine, there are plenty of normal organisms that live there. And then finally we have the genital tract, and typically I'm talking about the genital tract of females. That usually grows more normal flora than the genital tract of males. Males grow normal flora, but that is the flora that lives on their skin around the genital tract area. There are a number of different factors that determine the composition of your normal flora depending on what body site the normal flora is in. One factor that helps determine the composition of your normal flora would be the nutrients that are available at the site. Also environmental conditions, so what is the degree of humidity or moistness around the area where we're trying to grow normal flora. Oxygenation, so in other words, anaerobes prefer an environment with no oxygen, and aerobes prefer an environment with oxygen. If you take a look at the number of anaerobes per number of aerobes in the gut, they actually outnumber aerobes 1,000 to 1. And then there are substances like bile salts, lysozyme, or fatty acids that are in the mouth or on the skin, and those things can affect your normal flora. A lot of organisms cannot tolerate bile salts, and then other organisms have their cell walls lysed because of lysozyme, and even fatty acids can be antimicrobial. And then finally, we have to consider the pH of the site. For example, the pH of the stomach is about 1 to 2, which is very, very acidic. And so most organisms are not going to survive the stomach. However, Shigella is kind of a misnomer. It can actually survive the stomach. Okay? An organism's ability to establish itself successfully in any given body site depends on its ability to survive and thrive in the body site that it actually colonizes. We have about 100 trillion organisms that live on or within us at any given time, and of these 100 trillion organisms, we harbor hundreds of different species of organisms. In fact, about 3 pounds of our body weight is attributed to the bacteria that we have on or within us, and 25% of all human deaths are attributed to some sort of infectious disease. And the infectious diseases that I most commonly think of would be things like malaria. That's caused from an organism known as Plasmodium. E. coli 0157H7 is another organism that can cause hemolytic uremic syndrome and all kinds of problems in the gastrointestinal tract. And then other organisms that are pretty highly responsible for human deaths would be Mycobacterium tuberculosis. So that's 3 examples of organisms that cause infectious diseases. Now, like I said, we do have normal flora or normal resident flora that lives on the skin, and those organisms can tolerate the conditions of the skin, including lipids on the skin, and they can tolerate those fatty acids. Actually, if you don't have fatty acids in your diet, your skin can crack, and then some of those organisms can become pathogens. Okay? Now, many organisms prefer the moistness of the skin, so you do want to keep your skin hydrated. And there are a number of common indigenous microbiota of the skin, including coag-negative staphylococcus. We've got alpha and gamma streptococcus. We've got diphtheroids, which are also known as Corynebacterium species. We've got Propionibacterium, which is now called Cutibacterium. And then we have certain types of yeast, like Candida, for example. These are all examples of normal flora of the skin. Now keep in mind that 90% of this flora can be temporarily removed by a good hand washing. And you do want to wash your hands and scrub your hands, because that soap with the friction can actually get rid of about 90% of that skin flora, at least temporarily. Let's talk about the normal flora of the upper respiratory tract, and that includes the mouth and the nasopharynx, okay? The bronchioles, the lungs, and the bronchial alveoli, those are all part of the lower respiratory tract. Okay, and the lower respiratory tract should be relatively sterile, but the upper respiratory tract is colonized with normal respiratory flora. And the organisms of the upper respiratory tract are able to tolerate oxygen and CO2 exchange and mucus. Okay? Some common indigenous microflora of the upper respiratory tract would be things like alpha and gamma streptococcus, lactobacillus, and lactobacillus is a normal gram-positive rod that can live in the upper respiratory tract. It can live in the gut. It can also live in the genital tract. Staphylococcus, including coag-negative staphylococcus, miscellaneous anaerobes, and Neisseria species. Now, this does not include Neisseria gonorrhea, but it does sometimes include Neisseria meningitidis. About 30% of all people in the world are colonized in the nasopharynx with Neisseria meningitidis, but also Neisseria meningitidis, the other 70% of the time, can be a pathogen. So those are organisms that are common indigenous microbiota of the upper respiratory tract. And there's a number of different normal organisms that live in the digestive tract as well. And these organisms can tolerate a lower pH and lower levels of oxygen. Like I said, anaerobes outnumber aerobes 1,000 to 1 in the digestive tract. So common indigenous microflora of the gastrointestinal tract would include your enterics, the Enterobacteriaceae, which is a big family of gram-negative organisms. And some examples of the enterics or the Enterobacteriaceae would be Escherichia coli, Klebsiella, Proteus, Providencia, Morganella. Those are just a few examples of normal enterics. Then we've got Enterococcus, which is basically a strep of the gut. Entero means of the gut, and coccus means cocci. So this is a streptococcus of the gut. This is called Enterococcus. Then we have Staphylococcus, and we have miscellaneous anaerobes. That could be any number of anaerobes, but these are all examples of common gut flora. Then finally we have the normal flora of the genital tract, and most of the time I'm talking about normal flora of the female genital tract. Flora that colonize around the penis of males, that is actually going to be skin flora. But genital tract normal flora around the vagina would include things like lactobacillus, Staphylococcus, Streptococcus, diphtheroids, and miscellaneous anaerobes. One such example would be Mobiluncus. Okay. Other examples would be gamma streptococcus or enterococcus can be normal flora of the genital tract if it's only a few colonies. Staph aureus can be normal if it's not in a pure culture. You got to be careful though, because Staphylococcus aureus can actually cause toxic shock syndrome as well because of the toxic shock syndrome toxin. But Staph aureus can also be normal in the genital tract, so it just kind of depends. We've got to weigh that out. Here's a few more terms for you. The first one I want to talk about is pathogenicity, and this is basically an organism's ability to invade the host tissue, alter the host's health, and cause disease. That's a microbe's pathogenicity. The degree of pathogenicity of an organism is called its virulence, okay? So a virulence factor is something that makes something more pathogenic, okay? This is a property that causes the organism to produce a disease in its host. There are several virulence factors. I'm going to give you a few examples. If the organism has a capsule or pili, or if the organism produces toxins or enzymes, these can all be different virulence factors causing the organism to be more pathogenic. And then we have the opportunistic organisms, which are organisms that are usually part of the normal flora, but under certain circumstances can cause disease. So a great example of that is Streptococcus pneumoniae. Again, it lives in the upper respiratory tract as normal flora, coexisting with other organisms that are normal flora. But for some reason, if you're elderly, if you have a decreased immunity or an immune system that isn't functioning correctly, that Streptococcus pneumonia can actually cause disease. Opportunistic organisms or opportunistic pathogens usually have some sort of trigger that makes them more virulent. For example, their habitat could be changed. For example, if you have an organism that lives in the gastrointestinal tract, if that gastrointestinal mucosa is damaged, like a skin tear or something like that, then all of a sudden normal organisms can be opportunistic organisms. If you have their habitat that is disturbed or changed due to pH or antibiotics or something like that, that can actually cause an organism to be an opportunistic pathogen. Okay, in the host itself, if the host is immunosuppressed, if their immune system is weakened or compromised, you're more likely to develop an opportunistic infection. So how does an organism actually cause infection or disease? Okay, usually there is some competition between the host immune system and a potential pathogen to start out with. Okay, the pathogen has to have some special ability to overcome the host defenses in order to cause disease. So they have to have special abilities such as virulence factors. Okay, there's actually 4 disease stages in the progression of an infectious disease. So 4 stages in the disease process would be things like the incubation period, the prodromal phase, the acme period, and the period of decline. So in the incubation phase, your patient is sick and contagious, but they may not know it yet. A lot of times during the incubation period, our patients are asymptomatic. In the prodromal phase, signs and symptoms have started to occur. Usually these signs and symptoms are vaguely apparent and your patient is feeling vaguely under the weather. In the acme period, this is the peak of signs and symptoms. In this stage, treatment like antibiotics and antiviral medications are usually given, or if you're very sick during this stage, it could be with a self-limiting illness. And then finally, the 4th stage of the disease process is the period of decline, and that's where your symptoms start to cease, and the infection is actually brought under control during that period of decline. So two notable virulence factors of bacteria would be things like enzymes that the bacteria produce or toxins that they produce. So on this slide, what I've done is I've just given you a few examples of enzymes and toxins that are going to be virulence factors for bacteria. Okay, so anytime a word ends with "ase," it usually means that it's an enzyme. So for example, Staphylococcus aureus makes coagulase. Coagulase forms clots that stick to an organism so it can't be phagocytized. So that enzyme made by Staphylococcus aureus is a virulence factor. Streptokinase and hyaluronidase are both made by Streptococcus pyogenes. Streptokinase dissolves clots, allowing the pathogen to spread easier, and hyaluronidase actually breaks down the tissue cement, allowing bacteria to spread easier as well. And then finally, some organisms like Staph aureus produce leukocytins, and leukocytins actually destroy neutrophils and macrophages. So that, if you remember anything from immunology, that limits our phagocytosis. Okay. Other virulence factors produced by bacteria would be toxins. Some notable toxins would be the botulinum toxin produced by Clostridium botulinum. This is a neurotoxin that affects the nerves. We've got the diphtheria toxin from Corynebacterium diphtheriae, which actually inhibits protein synthesis in humans. And then we have other miscellaneous toxins that either stimulate fever, hypotension, or circulation, etc. For example, Staphylococcus aureus, you can get a bad toxin infection from enterotoxins A and D from eating bad fried rice that's been sitting in the refrigerator for a long period of time. So those are some examples of virulence factors of bacteria, the enzymes and the toxins. Now, there are a number of specific roles of normal flora. There are actually things that they do to help us, okay? For example, normal flora stimulates antibody production in the host. Normal flora also competes with pathogens for nutrients, keeping the pathogens suppressed. Normal flora helps in the synthesis of vitamins like vitamin K. They aid in normal metabolism. For example, bacteria break down bilirubin to urobilinogen. So those are examples of why normal flora is beneficial to the host. Now, there are ways as the host that you can resist certain types of pathogens. You can have a physical barrier, like the skin is a physical barrier, and components of the skin are physical barriers, including things like lysozyme or the sloughing off of epithelial cells, or sebum, which is skin oil. Those are all physical barriers. Okay, we have cleansing mechanisms, which would be sloughing off of the epithelium, tears, mucous membranes, and mechanical cleansing during urination. Those are all ways of removing pathogens from the body site where they are living. And then we have antimicrobial substances, including things like lysozyme, beta-lysins, and the antibody secretory IgA, which protects your mucous membranes. Other host resistance factors would include things like indigenous microbial flora. So some other types of bacteria produce bacteriocins. So this is normal flora that produce bacteriocins, and bacteriocins secreted by some bacteria, it actually inhibits the growth of closely related pathogenic or opportunistic organisms as well. In phagocytosis, we have neutrophils, monocytes, and macrophages that are drawn to the site of infection by a chemical signal. That's called chemotaxis. They attach to the organism and become a phagosome. They ingest that organism and kill it with cytokines like hydrogen peroxide, myeloperoxidase, and a number of other types of enzymes inside of the white cells. We have inflammation. Inflammation actually causes our pathogens to be eradicated. And then finally, we have our natural or adaptive immunity. So we've got active immunity and passive immunity. We're either born with our immunity, or we can actually gain immunity from something like a vaccine. Epidemiology is the study of how infectious diseases spread in a community. It's the study of outbreaks and disease spreading, and this is directed by federal, state, and local health departments usually. Okay, the route of transmission for disease transmission would include several different things, including inhalation, ingestion, close contact, or injection. Okay, and I'm going to outline these all more in the next few slides, but inhalation is when you are in the line of fire when it comes to airborne and droplet transmission. Ingestion is when you've eaten either contaminated food or drank contaminated water. Close contact disease is usually something that is sexually transmitted or when you come in contact with secretions or body fluids. And then finally, injection is when you're punctured by something infectious or you have a cut or a bite by something that's infectious. So let's go ahead and talk about all of these routes of transmission on the next few slides. So inhalation is one way you can be susceptible to getting a disease. We can actually reduce airborne transmission in microbiology by the use of biological safety cabinets. Okay, this inhalation is also known as airborne or droplet transmission, and usually you become sick by coming in contact with somebody that's been sneezing, coughing, or talking. Okay, transmission of both viral and bacterial diseases occurs by inhalation. Some examples of bacteria and viruses that you can get through an inhalation pathway of airborne and droplets would be things like influenza virus, rhinovirus, that's what causes the common cold, and you can actually come in contact with bacterial infections such as Streptococcus pneumoniae and Mycobacterium tuberculosis. Also strep pyogenes, which is the organism that causes strep throat, you can get that through inhalation as well. Another way that a host can come in contact with an infectious disease is by ingesting it. Okay, a lot of times what has happened is that the patient has come across something via the fecal oral route, and what that means is that they've either eaten something or drank something that has been contaminated with human or animal feces. Okay, when a person gets an ingested type of disease, the damage of there is damage to the lining of the GI tract by toxins or invading, or those organisms invade the tissues. Okay, some bacterial toxin producers would be Clostridium botulinum, Bacillus cereus, and Staphylococcus aureus. Now, viruses and parasites can cause ingestion diseases as well. Like an example of a parasite would be Giardia lamblia. An example of a virus would be norovirus. Those are some good examples. Close contact is another way that a host can become sick with an infectious disease. And a lot of times that means through kissing or salivary glands, like that's how people can get infectious mononucleosis, or skin and genital contact. That could be hepatitis B, that could be HIV. Those are things that you can get through close contact. Okay, the herpes simplex virus and the Epstein-Barr virus, which causes infectious mononucleosis, those are transmitted through salivary transmission. And then human papillomavirus, syphilis or treponema pallidum, chlamydia, trichomoniasis, Neisseria gonorrhea, and HIV. These are all types of diseases that you can get through genital or skin-to-skin contact. And then finally, I just want to mention that chlamydia is the most common sexually transmitted disease in the U.S., with about 900,000 diagnosed cases a year. The last way that a host can be exposed to an infectious disease is through injection. And when I think of injection, it's not just needle sticks. It could be a needle stick or from broken glassware, but it could also be a bite wound or a sting from an insect or an animal. Okay, so some infectious diseases that come from injections would be things like Plasmodium, that is malaria, Pasteurella multocida, which is flora of dog and cat mouths, Eikenella, which harbors human mouths, and then HIV and hepatitis B, which are viral and blood-borne infectious organisms. All right, so it's time to switch gears a little bit. Now we're going to talk about the impact of microbes on the environment. Now, microbes on the environment, they either have a positive effect where they are beneficial to the environment, or they can have a harmful effect where they actually cause harm on the environment. So there are probably many more bacterial beneficial roles than there are harmful roles. So bacteria breaks down dead things, okay, as they're carnivores, they break down dead things, and fungi break down decaying vegetation, okay. So we could say that fungi are saprophytic organisms, and that just means that they eat dead and decaying vegetation. The metabolic activities of bacteria affect plants and animals. And then finally, bacteria is useful in food production and biotechnological processes. Okay. The harmful roles of bacteria and viruses is that they can contaminate food and water. Okay. We could actually ingest something that has been fecally contaminated. Okay, and if we are eating unsafe food or water, it's basically causing our host a disease. So that's a harmful role of microorganisms on the environment. There's a number of microbial benefits. Bacteria in the intestines of animals digest nutrients and produce vitamins and growth factors for humans. So actually bacteria play a role in the synthesis of all kinds of things like thiamine, which is an amino acid, vitamin K and vitamin B. The bacteria plays a role in the synthesis of all of those types of amino acids and vitamins. Okay. Microbes can be used in the production of foods such as wine, yogurt, sour cream, cheese, and even things like soy sauce, black teas, and pickles. Microbes can use fermentation to convert corn sugars to ethanol. So corn fermentation equals ethanol, but cyanobacterium equals butanol, which is a cheaper, more efficient way of making fuel. Okay, microbes can be used in the pharmaceutical and medical industry. So for example, we can use penicillium, which is a mold, to make penicillin. Okay, microbes can be used to make vaccines. So the diphtheria toxoid, which is a detoxified toxin, is used to immunize people against the disease diphtheria. Other beneficial roles of microbes, they are useful in biotechnology, and they play a major role in genetic engineering. So we can produce masses of hormones, enzymes, and antibodies for medical treatments. And then we can use microbes for general research. For example, E. coli has been poked and prodded every which way so people could study DNA replication and protein synthesis and a number of other things. So those are the benefits of having microbes in our world. Now, like I said, microbes can be not beneficial to humans as well, or not beneficial to the environment. Okay. For example, there are about 1 billion people that lack access to safe drinking water. That's 1/7 of the world population. And if you look at places like Afghanistan, Chad, and Mali, these are places where very few people in the population have access to clean drinking water. Okay, unsafe drinking water has contributed to 7 cholera pandemics, and cholera is produced from a pathogen known as Vibrio cholerae. But those 7 pandemics included one in India in the early 1800s, one in India and Russia that was in the mid-1800s, and then in the late 1800s there was the India, Asia, European, African, and North American cholera outbreak. In the early 1900s, there was the India, Middle East, North African, Russian, and European. I guess that would be an epidemic. And then finally, in 1961 to present day, they say that we're in the midst of the 7th cholera pandemic. Okay? Let's go ahead and take a comparison of good unpolluted water versus bad water or unhealthy polluted water. Okay? In unpolluted water, there are no carbon structured dead things in the water. Okay? Unpolluted water is low in organic compounds. The amounts of bacterial species that are present in the water is kind of limited. And most organisms that are in the water come from runoff. So some examples of organisms that are in the water that is unpolluted would be Actinomycetes, Cellulomonas, Paramecium, Amoeba, and Euglena. And in polluted water is high in organic compounds from sewage, feces, and chemicals. There are many bacteria coliforms that exist in contaminated water. Okay, a coliform is basically a fancy word for a mixed bag of organisms found in the gastrointestinal tract of humans and other animals. If you have a coliform that's in your water, it indicates some sort of fecal contamination. Okay? These types of organisms like E. coli and Enterobacter dramatically affect the ecology of the water. Phosphates start to accumulate, giving rise to algae blooms, and algae blooms provide nutrients for other microbes that rapidly reproduce using all available oxygen. Animals and plants will die, and anaerobic bacteria thrive in the muddy bottom. So those are examples of unpolluted water versus polluted water. So water pollution comes in three types: physical pollution, chemical pollution, and biological pollution. Okay? Physical pollution is when you have sand or soil that gets into the water. Chemical water pollution is when you have copper and acids and other types of chemicals that get into the water. And then biological pollution is when you have fecal contamination that occurs in the water. Okay? So a physical example of water pollution sources would be phosphates from soil runoff. Chemical examples would be spills of chemicals and pesticides. And biological pollution in water would include things like bacteria from human and animal feces. As you can see here, the number of organisms that can cause water pollution is several. Okay, so there's a number of bacterial reasons why water could be contaminated, including salmonella, which can lead to typhoid fever, Vibrio cholera, which can lead to cholera, Shigella, which can lead to shigellosis, and then Campylobacter species. Okay, and then we have viral things that can cause water pollution, including hepatitis A, Coxsackie virus, which is an enterovirus, causes mild flu-like symptoms, and then norovirus, which is also known as the winter vomiting disease, which is the most common cause of all cases of stomach flu. Then we have parasitic reasons why water can be contaminated, including Entamoeba histolytica, Giardia lamblia, and finally Cryptosporidium. Cryptosporidium is pretty uncommon. It's an opportunistic parasite that can cause diarrhea, especially in immunosuppressed people. For example, in Milwaukee, Wisconsin, there was an outbreak in the late 1990s. There were 450,000 people that were infected with Cryptosporidium, and there were 100 deaths or so because of a contaminated water supply. And the people that passed away from this Cryptosporidium outbreak were mostly people that were immunosuppressed, like HIV patients. Now, there are some bacteria that are considered to be nitrogen fixers or denitrification organisms, and one such example would be Rhizobacterium. Okay? The populations of microbes in soil vary according to the soil type, and bacteria are often the most abundant inhabitants of soil. There can be fungi, algae, and protozoal in the soil as well. Some fungi that is present in soil would be Penicillium, Aspergillus, or Fusarium. Now, viruses do not live freely in soil, but they live within organisms in the soil. Okay, so kind of keep that in mind. Now, microbes can live in both freshwater and saltwater. Usually they live as biofilms on surfaces, and a biofilm is a community of organisms living together that communicate and act as a whole. Okay? Saltwater sources have more nutrients, thus there are more microbes in saltwater. Freshwater sources have less nutrients if uncontaminated, thus they have less microbes. And water source contamination with sewage contamination harbors a large population of microbes. Okay, there are good organisms in water, including Monera, which is a kingdom that includes bacteria. Protists are also good organisms that live in water. That would be things like Paramecium and the eukaryotes. These things, Monera and Protists, break down organic matter. The good bacteria remove organic matter, remove ammonia and nitrates, and remove phosphates from the water. So like I said, when there's a natural disaster, the microbe could take over and you could get an opportunistic infection because of a natural disaster. So we have to keep that in mind. So here's some examples of natural disasters that have led to bacterial kind of pandemics, okay? So floods. In a flood situation, the microbe numbers may not increase but are more easily transmitted from person to person. So for example, I talked about the Yellow River in 1931 when there was up to 3 million people dead. The Yellow River is in China. And basically, 88,000 kilometers were flooded, and there was people that drowned, and then there was famine because of this flood, and also a lot of disease. The Yellow River in China is nicknamed China's sorrow due to this disaster. Tsunamis, you have similar issues with tsunamis and floods that arise from floods. Infectious disease is communicable due to contaminated water, lack of shelter, and lack of medical care. And the tsunami that I think of most is the Indian Ocean tsunami that happened in 2004, where there were 300,000 dead people due to cholera, typhoid, malaria, and dengue fever. Because you can see tsunamis causes the water to rise. With rising water, you've got mosquito infestation. With mosquito infestation, you've got more transmissions of dengue fever and malaria. Okay. The third largest earthquake recorded ever was the one that preceded the tsunami in 2004. This earthquake was recorded at a magnitude of 9.1, and that's what led to the tsunami in 2004. Earthquakes can lead to serious problems with microbes. Disease occurs from contaminated water, lack of refrigeration, and vector-borne illnesses from standing water. And here we've got the same situation in the Indian Ocean in 2004 where there were 300,000 people dead. Okay, and then finally we have hurricanes where contaminated food and water start to occur. Shelters become crowded and vector-borne diseases start to go up. And what I think about here are the cyclones and hurricanes that happened in 1970s in Pakistan, India regions, and in Bangladesh. Okay, there were 500,000 people dead, which is kind of hard to fathom. If you compare the hurricanes in Bangladesh in the 1970s to the Katrina hurricane in the United States in 2005, there was only 2,000 deaths that occurred in the Katrina hurricane in 2005, which is pretty crazy when you compare that to the Bangladesh hurricane from the 1970s. Chemicals are transformed via enzymes and life forms to sustain life. Amino acids, carbohydrates, and lipids make up cellular structures. In metabolism, these cellular structures are either made or they are broken down for energy. So let's go ahead and review a couple terms that we talked about before. One is called anabolism, which is the building of amino acids to make cellular structures. And the other term that I want to re-familiarize you with is catabolism, which is the breaking down of carbohydrates in order to make energy. There are certain cycles essential for life that have to happen in the environment. And the cycles that I'm going to outline today are the carbon cycle, the sulfur cycle, and the nitrogen cycle. Okay? In the carbon cycle, there is a certain supply of carbon in nature, and that is limited. Okay? This is called the laws of thermodynamics. Okay? Carboxidobacterium, which is an autotroph, are organisms that are able to oxidize carbon monoxide to carbon dioxide using the enzyme carbon monoxide dehydrogenase. Okay? Heterotrophs consume autotrophs and catabolize organic molecules for energy. Okay? So the carbon cycle is definitely influenced by carboxidobacteria, which oxidize carbon monoxide to carbon dioxide using an enzyme. The carbon cycle and carbon molecules are crucial for life on Earth. Carbon affects the atmosphere, biosphere, the oceans, and human plant life. Okay? Carbon is constantly being recycled by bacteria and plants and the consumption of fossil fuels, but each trophic level gets less heat units with each. That's called the laws of thermodynamics. The next cycle influenced by microorganisms in the environment is called the sulfur cycle. Sulfur is needed in the formation of amino acids because amino acids form proteins. Bacteria release sulfur from hydrogen sulfide, making sulfates, and now the sulfates are available for use to plants, animals, and humans. Some sulfur oxidizers are bacteria species, and they oxidize sulfide as a source of energy. Sulfur oxidations is when you have microbes that convert hydrogen sulfide to elemental sulfur or sulfates. Hydrogen sulfide is actually toxic to life, and it must be reduced to sulfates or elemental sulfur. And we need sulfates or elemental sulfur to make amino acids and proteins. Okay? H2S producers oxidize sulfur, leading to sulfates. The last cycle I'm going to talk about is the nitrogen cycle, and organisms play a role on the nitrogen cycle as well. Nitrogen is essential in making of nucleic acids, and that would be things like DNA and RNA. Okay. Nitrogen fixation is when nitrogen gas is reduced to ammonia by some bacteria. Only bacteria will fix nitrogen. Ammonia can be used by plants and animals. Ammonia is used by animals as a precursor for the formation of amino acids in nucleotide synthesis. Ammonia is converted to nitrates and nitrites by bacteria. This is called nitrification. And nitrates and nitrites are in the atmosphere. Okay, nitrates is NO3 and nitrites is NO2. Nitrogen fixation traps this atmospheric nitrogen, which can be used by plants when it's time to be replenished into the soil. Plants make amino acids and then proteins, which are essential for human life. Without plants, nitrogen will be unusable by humans. The final products of the nitrogen cycle become the building blocks for amino acids, purines, and pyrimidines. And if you remember from previous lectures, we need purines and pyrimidines to make DNA and RNA. So here's an overview of the nitrogen cycle, and we're going to talk more about this in lab when we work with nitrogen fixers in organisms that do denitrification and ammonification. But bacteria fix atmospheric nitrogen gas. They form ammonia from nitrogen gas. Bacteria use some of that fixed nitrogen, and the remainder goes back into the soil. The nitrogen fixers die, and then nitrogen is recycled by other organisms. So nitrification is a two-step conversion of ammonia to nitrite and then to nitrate. Denitrification is when bacteria return nitrogen back to the atmosphere as nitrous oxide or nitrogen gas. Let's talk a little bit about industrial microbiology and biotechnology. So it's not limited to just alcohol fermentation or cheese production, but that's probably the first things that we think of. We can actually use industrial microbiology and biotechnology to make things like acetone. And when we make acetone, we can use that for things like gunpowder. It's the metabolic reactions present in microbes that allow bioengineering to occur. Okay? Microbes have many enzymes that can convert miscellaneous chemicals, and this can happen quickly because of the rapid metabolism of these organisms. Think of generation time, the time it takes for a microbe to produce an identical genetic copy. For example, Escherichia coli, it doubles every 15 to 20 minutes. Which of course would make it an excellent microbe for bioengineering. Now we can use bacteria for biotechnology and industrial microbiology, and what we do is we take the bacteria and put it in a large container known as a bioreactor. In the bioreactor, the temperature, pH, oxygen, and nutrients are all controlled. Okay? Biotechnology is when we use bacteria or microbes to contribute to the health or improve the quality of life and interrupt the cycle of disease. So one example of biotechnology is the making of a pluot. I don't know if you've ever eaten a pluot, but they are absolutely delicious, and they are a cross between a plum and an apricot. Okay? Some products of biotechnology would be enzymes, hormones, antibiotics, and antibodies. These are all products of biotechnology. And then this slide basically outlines some examples of the microbial sources in the commercial markets and how they can benefit you. So in the dairy industry, we can use bacteria to to take lactase and proteases to hydrolyze whey in dairy products to make cheese. Okay? So eating cheese, of course, is a wonderful thing. In the brewing industry, molds and bacteria use cellulase and amylase to liquefy and clarify the mash. I don't know much about making wine or beer, but it sounds delicious. Meat tenderizing. We can actually use bacteria or molds and proteases to tenderize meats or lipases that break down fats in lunch meat. We can actually use bacteria to make cleaning supplies. Some bacteria and molds use amylase, cellulase, and lipases to break down stubborn stains. Okay? And then finally, another example of bioengineering is paper milling, where we can use molds and yeast and xanthanases to break down the pulp of trees for paper processing. So that's a pretty good day. You're eating cheese, you're drinking wine, you're eating a few chunks of salami. Of course, you might spill your wine, so you can clean it up if it gets on your shirt with a little bit of the cleaning supply stuff from the cellulase and amylases. And then finally, you can do all of this while reading your poetry on the paper that's been milled by bioorganisms. Now, antibiotics are also the result of bioengineering. It's very ironic that we can use mold and bacteria to actually inhibit or kill other molds and bacteria. We can actually hijack the biosynthetic machinery of bacteria, according to a study conducted at Stanford University. We can make or take things like penicillium, which is a mold, bacillus, and streptomyces, which are bacteria, and we can actually take that, the machinery of those bacteria and change it because it's highly malleable. And when we're changing that, we're basically genetically engineering things like vancomycin, erythromycin, and several other antibiotics, including the polypeptide antibiotics. All right, so that is it for Unit 4. Today we've talked about the interaction and impact of microbes on humans and microbes on the environment. You have a great day.